Hello, I'm Dr. Mirko Pegoraro, and this will be a lesson that is related to your second workshop. In this lesson, we are going to talk about the axis formation in the Drosophila melanogaster embryo, as well as the segmentation of the embryo. So, what are we going to do in this lesson? So, in this lesson, we are going to learn how the Drosophila embryo is formed in the anterior-posterior axis. We are going to learn about the different genes that form the segmentation network, and we are going to learn about the OMC genes, which we already talked about in the past lessons when we were talking about the formation of the anterior-posterior axis in mammals. This morning you had a lesson with Dr. O'Reilly about the neural cell development. The next lesson next week will be about the paraxial mesoderm, and this lesson is linked to the Drosophila melanogaster DPP expression practical, the imaginal disc practical, and the metamorphosis and regeneration lesson. So, the anterior-posterior axis formation begin already at the oogenesis in Drosophila melanogaster. Oogenesis, just a reminder for you, is the formation of the egg. So we are already talking about axis formation during formation of the egg inside the female ovaries. We will see that there is a hierarchy of genes that act in a hierarchical manner to establish the anterior-posterior axis, or the anterior-posterior polarity, if you so want, and also divide the embryo in different functional segments. So the first genes that are activated in the embryo are the maternal genes. The mother, during the formation of the egg, actually deposit mRNA, messenger RNA, inside the egg, and already in depositing this mRNA, we have the formation of two poles, the anterior and the posterior poles. This maternal mRNA encode for transcription regulator or transcription factors, if you wish. So this mRNA, when translated into protein, produce transcription regulators or factor, and these regulators control the expression of the first zygotic genes transcription. So these maternal genes, maternal mRNA that are translated into proteins, into transcription factor, control the expression of the zygotic genes. What are the zygotic genes? The genes that belong to the zygote after fertilization. Okay? The first genes that are expressed in the zygote are the GAP genes. The GAP genes are expressed broadly along a large part of the embryo, and they overlap to each other. And they are the first genes that start to establish segments in the embryo. After the GAP genes, we have expression of the pair rule genes. In fact, the GAP genes directly control the expression of the pair rule genes along the anterior-posterior axis and divide the embryo in 7 expression bands, which overlap more or less to physical bands on the future larva. The pair rule genes control the expression of segment polarity genes. These genes divide the embryo transcriptionally in 14 segments. These 14 segments correspond approximately to the 14 segments of the anatomical segments in the embryo. Finally, the GAP genes, the pair rule genes, and the segment polarity genes interact with each other to coordinate the expression of the homeotic selector genes. The homeotic selector genes are master morphogens and control the morphogenesis of the different segments of the developing embryo. So one segment will develop legs, one segment will develop wings, some segment will become head, and so goes on. Whether the segments become different anatomical parts depend on the expression of the homeotic selector genes. So let's have a look on how the anterior-posterior axis is established in Drosophila already at the level of oogenesis. So here you have in this cartoon an example of a growing oocyte, so in yellow. So the oocyte now is inside the maternal ovaries. So we are looking inside the maternal ovaries and what's happened to the developing oocyte. So we find there that a gene called gherkin is expressed, or perhaps better would be to say that the nurse cells, which are cells that support the growing egg, express this mRNA and deposit this mRNA inside the developing oocyte. Inside the developing oocyte, this mRNA is translated into a protein, the gherkin protein. The gherkin protein is localized specifically in between the nucleus, which is that blue dot that you see in the middle of the yellow cell. So gherkin is localized between the nucleus and the posterior follicle cells. In fact, gherkin bind the posterior follicle cell receptor torpedo, causing posterization of these follicle cells. Other mRNA are deposited by the mother, by the nurse cells, inside the developing oocyte and are transported in different regions of the oocyte. For example, Oscar and Nanos mRNA are deposited inside the developing oocyte and are transported posteriorly, while bicoid is transported anteriorly. During the maturation of the oocyte, at one point, the nucleus from a posterior position migrates toward an anterior position and a dorsal position. And it does so carrying a cargo of the gherkin protein. These gherkin proteins that are around the nucleus while it's moving interact with the follicle cells in the dorsal part of the developing oocyte, causing dorsalization of those cells. So gherkin is important to establish an anterior-posterior axis very early during the oocyte development, but also to establish a dorsoventral axis quite early during the development of the egg. Now, in a mature egg, we have the nucleus in a dorsal anterior position. We have bicoid mRNA concentrated in an anterior position and the nanos mRNA concentrated in a posterior position. You can see here a mature egg. So this is a mature egg, so it's not a fertilized egg, so it's not a zygote. In a mature egg, we find the maternal pronucleus in a position that is dorsal and anterior. We have a concentration of bicoid mRNA in an anterior position, and we have nanos mRNA concentrated in a posterior position. After fertilization, so when the egg is fertilized and then released from the maternal body, so it is laid, the nucleus of the zygote start to divide, and the different nuclei actually do not divide the cells. Cytoplasm. In fact, they share the same cytoplasm and they migrate all around the peripheries of the embryo. You can see here all those circles represent the different nuclei during the cell division, during cleavage. They migrate around the periphery of the developing embryo, but they are sharing their cytoplasm. They are not individual cells per se at this stage of development. Because of this, the bicoid mRNA that is translated into bicoid proteins and the nanos mRNA that is translated in nanos protein can diffuse. So the proteins produced by these two mRNA, the nanos and bicoid proteins, can diffuse from a posterior position toward the anterior and from the anterior position toward the posteriors. So bicoid proteins form a anterior to posterior gradient, and the nanos protein form a posterior to anterior gradient. So the different cells now are able to use these two opposing gradient of proteins as a GPS, so as a way to understand where they are along the anterior-posterior axis. So if they are exposed to a lot of bicoid protein, the cells become anterior cells. If they are exposed to a lot of nanose proteins, the cell become posterior cells, and so on. On other two mRNA that are extremely important for the establishment of the anterior-posterior axis during early development are hatchback and caudal mRNA. And here we are talking about, at the moment, the oocyte, so the forming oocyte. So the mother deposits bicoid and nanos, as well as hatchback and caudal. However, contrary to nanos and bicoid, hatchback and caudal mRNA are found at the same concentration along the anterior-posterior axis of the developing egg. So how it is possible now that mRNA that is concentrated the same along the anterior-posterior axis are important for an establishment of an anterior-posterior axis in the embryo? Well, it is because of a process called repression of translation. Bicoid protein, which you remember is produced mostly anteriorly because the mRNA is concentrated anteriorly, repress the translation of caudal mRNA. So caudal mRNA cannot be translated into caudal protein anteriorly where we have a lot of bicoid protein. And it can only be translated into caudal protein in a posterior position where we don't have much of bicoid protein. Conversely, nanos protein repress hatchback mRNA translation. So hatchback can only be translated anteriorly, and so we found the protein anteriorly, but not posteriorly, where we have a lot of nanos protein. So, because of the concentration of bicoid mRNA and nanos mRNA in the anterior and posterior position, respectively, and because they then become protein, are translated into protein, mostly anteriorly bicoid and mostly posteriorly nanos, these two gradients also control the translation of hatchback and caudal anteriorly in a posterior position. At this point, the zygotic genes can be activated. What are the zygotic genes again? So when the egg is fertilized, it forms a zygote. So the zygotic genes are the genes that belong to the zygote. Okay? So at one point, the zygotic genes are activated, and in fact, the maternal gene control the activation of specific zygotic genes in a specific order. The cell fate of the developing embryo are determined by the segmentation genes. The segmentation genes are the genes that are activated after zygotic activation. So when the genes of the zygote are activated, they are the first ones that are activated in a sequential way, and they control how the embryo forms different segments along the anterior-posterior axis, and in this different segment, different cells express different genes. So the first genes to be activated after zygotic transition are the gap genes. The gap genes, here you have an example of the pattern of expression of one of the gap genes, which is called Kruppel, and you find it expressed in a middle region in the early embryo and in several segments in a later embryo, which correspond to several segments of the larva. So these gap genes are called so because if you mutate them, the larva lose a lot of continuous genes. So these gap genes are expressed in several continuous segments. So if you compare here this larva on the left with the small larva on the right, the small larva on the right are missing all these purple segments where we have expression of Kruppel. So if you remove the expression of Kruppel, we lose all those segments. The gap genes control another layer of segmentation genes, which are the pair rule genes, and the pair rule genes are expressed in any other segment. So you can see here the expression of Fujitarasu. You can see that it's expressed not in all the segments, but in any other segment. And if mutated, so if we remove Fujitarasu, we lose a portion of not all the segments, but any other segments. And finally, the pair rule genes control the segment polarity genes, and the segment polarity genes are expressed in every single segment. So here you have a pattern of expression for a gene called engrailed, and engrailed is expressed in any single segment. And if we mutate engrailed, so if you remove the expression of engrailed, you obtain a larva which are missing a portion of any single segment. Okay, so the gap genes are directly controlled by the maternal genes, by the maternal mRNA, but soon after that they are activated, they form self-sustained feedback loops that control each other expression. And here you have a nice example of which genes control what. So the gap genes, after activation, form an anterior-posterior network of activation expression that sustain each other. For example, I'm just giving you like a few examples here of how these genes interact with each other. So anteriorly, we know that we have expression of hatchback, and actual repressed expression of knirps. Knirps is therefore expressed only in a more central posterior way, because in the anterior position we have highly expression of hatchback. Hatchback repressed expression of knirps. Hatchback also controls the expression of crapple. However, it controls the expression of crapple in a very peculiar way. High level of hatchback inhibit the expression of crapple. However, small level of hatchback are necessary for the expression of crapple. And therefore, crapple is expressed in a more anterior position compared to knirps. Because knirps cannot be expressed even if there is just a little of hatchback, while crapple need a little of hatchback to be expressed. So you see that the two of them, the pattern of expression of the two, are out of phase just a little. Hatchback and caudal induce the expression of giant. You can see here the expression of giant in green. So you find the expression of giant in an anterior and posterior position. However, in the middle, giant is repressed. It is repressed because in a middle position we don't have much hatchback nor caudal. In fact, giant in the middle is repressed by the expression of crapple. So where we have high level of crapple, we have repression of giant. So this kind of interaction between the gap genes are so precise that they can define the position of cells within a 1% margin error, which is much better than any GPS we can at the moment use. The gap genes, you can see here in the graph an example of different pattern of expression of gap genes. The gap genes control the expression of the pair rule genes. So these pair rule genes are expressed in any other segment, remember? So they are expressed in what are called zebra stripe. They have a zebra stripe expression. So they express in one segment, but not in the next segment, and then in the following one, and so goes on. Perhaps a very nice example of this pattern of expression are represented by even skipped and Fujitarasu. Fujitarasu we already encountered it. So even skipped is expressed in all the odd segments. So 1, 3, 5, 7, and so goes on. And is obviously skipping all the even segments. So 2, 4, and 6. Fujitarasu, on the other hand, is expressed in the even segment and not in the odd segment. We have 8 pair rule genes with overlapping expression. Each cells in a segment or a parasegment express, at the end of the day, a unique set of these pair rule genes. Okay, so we have now defined precisely different segment or parasegment, if you want, along the anterior-posterior axis. Parasegment are expression segment, so they are segment defined by expression of genes, which more or less overlap to anatomical segment, but they are out of phase by more or less about half of a segment. So a parasegment is out of phase compared to an anatomical segment by alpha segment. Okay, so inside a parasegment, or a segment if you wish, how the cells know whether they are in a more anterior or posterior position. So they acquire their anterior to posterior polarity inside a parasegment because of expression of winglets and hegemony. So cells that are in a more posterior position express winglets, while cells that are more in an anterior position inside a parasegment express hegemony. And this protein, when translated, form a gradient, a posterior to anterior gradient in the case of winglets, and an anterior to posterior gradient in the case of hegemony. So how these two genes are indeed expressed within the cells in a parasegment? So they are controlled by the expression of the pair rule genes. For example, engrail is important. Engrail is expressed in a more anterior position, while winglets is expressed in a more posterior position in the parasegment. And if we look on the expression of even skip and Fujitarasu, we can notice that only the cells that do not express neither even skip or Fujitarasu can actually express winglets. So the only cells that can express winglets are the cells that are in the posterior position in the parasegment. And therefore the cells that are in a more anterior position cannot express winglets, however express engrailed, and engrailed induced expression of hegemony. Now, all the segmentation genes control the expression, precisely end up controlling the expression of the homeotic selector genes in flies. We already talked about the homeotic selector genes before. These are genes that are important because they determine the morphology of each segment in the developing insect, in this case the fly. They are divided into complex, the Actinopedia complex and the bithorax complex, which are found in two different regions of the second chromosome, and in two different regions of the third chromosomes. So the Actinopedia complex are more anterior genes, and the bithorax complex are more posterior genes. And remember that their position in the chromosomes correspond exactly to their pattern of expression. This is called temporal collinearity. It's called temporal collinearity because the more anterior genes are expressed more anteriorly along the anterior-posterior axis, but also earlier during the development than the more posterior genes. Now, what do we find? In the head of the fly, the head of the fly is identified or determined by the expression of anterior genes like labia, deform, and proboscidea. Now, labia, of course, refer to the lips, if you want, proboscidea to the proboscis, so the trunk, if you want, of the fly, and then the form actually refer to deformation of the head, therefore it is expressed on the head. More posteriorly, we find sex comb reduction in Actinopedia, which tend to be expressed, which are expressed in the thorax of the animal, in particular the first and second segment of the thorax of the animal. On the third thorax of the animal is identified by ultrabithorax, and all the other abdominal segments are defined by expression of abdominal A and abdominal B. Okay, and this was the end of this lesson. We have learned today how the anterior-posterior axis is formed in Drosophila, as well as the different segments are formed and controlled by different hierarchy of genes, the segmentation genes. We should be able to recall and explain how the OMC genes are expressed along the anterior-posterior axis of the animal and are controlling the fate of different segments.